This podcast is all about the control of gene expression, specifically in eukaryotic cells. An average adult human is made up of like 30 something trillion cells. If you think about it, each type of cell has really specific jobs, like muscle cells, pancreatic cells, blood cells, neurons. If each cell carries the information for your entire genome, how does it know exactly what type of cell to be and how to do its job? Well, the answer to this is differentiation, which means cells only express a very specific combination of genes. So each cell becomes very specialized in its job, which is very important. So what we're going to talk about in this podcast is exactly how that happens. What's also cool though is differentiated cells can de-differentiate, meaning they can go from a specialized cell back to a cell that's not specialized yet. So almost like in the embryonic stage, but not quite once they're differentiated. So a lot of plants use this to clone themselves. They clone themselves before cloning was cool. If you have creeping Charlie in your yard, it's that like low weed with the purple flower. I have tons of it. It clones itself. So basically, like let's say you're mowing the lawn and you cut a chunk of the creeping Charlie off and it lands somewhere else. Those cells can actually de-differentiate and clone that plant, creating root cells and all new shoot and stems and leaves and all that good stuff. Starfish can actually do this as well. So if a predator grabs one of its legs, it can drop that leg and these cells will regrow a leg. Some starfish can completely clone themselves. It can actually, if a big enough chunk is cut off, create two new adult starfish, and they do that through de-differentiation. Gene expression is mostly controlled by deciding what genes will be transcribed into RNA. If the RNA is not made, then the polypeptide won't be made, the protein won't be made, and we won't show that phenotype. One of the big things that your cell does is it has these proteins called histones. You guys already know that chromosomes are 50% DNA, 50% proteins. Well, meet the histones. DNA is wrapped around each histone almost like you would wrap a shoelace around something. Then 8 histones are put together and DNA is further wrapped around those 8 histones, and that's called a nucleosome. When DNA is tightly wrapped around the histones, you can't copy it. Here's my analogy for it. If I have a notebook piece of paper and I write something on it and I ask you to copy this, you can easily do that. But if I wad up the paper, then you couldn't copy it. This is what your DNA does. This is your DNA wrapped around the histones. It can't be copied. This is your DNA relaxed, and it can be copied. That helps control gene expression. One of the coolest things that we've learned within the last decade or more is about epigenetics. So epi means above, and then genetics, so above the genome inheritance. What this is methyl groups attached to portions of your DNA. And these methyl groups will not allow that gene to be transcribed from DNA to RNA. Again, we're blocking RNA being made, which controls gene expression. What's really neat about the epigenome is it actually can be inherited, and studies are being done right now looking at those inheritance patterns. So what impact can epigenetics have? Well, believe it or not, these two mice are actually clones. As the embryos, they were implanted in mothers, and the mothers were both given BPA. BPA is a chemical commonly found in hard plastics, like in food containers. This mouse's mother was just given a normal diet, while this one's mom was given, folic acid, vitamin B12, all sorts of things that would be needed. What happened is in this yellow mouse, a gene was unmethylated, so it's always on. It's always accessible to be copied into RNA. And in the brown mouse, that gene was off. That chemical BPA causes the gene to be turned on. So basically, it removes that methyl group and leaves it on. So that's one of the things we learned about BPA that can be harmful, is it actually changes methyl groups. All right, let's check out an extreme example of DNA packing. So kind of like referring back to that idea of the DNA wrapped around the protein histones, this is even more extreme. So males have the sex chromosomes of XY, and if you recall, the Y chromosome is really small, and its main job is to tell the baby not to default to a female, but to go ahead and be a male. So each male cell only has one X chromosome, but females are XX, which means each cell has two X chromosomes. And we know that a duplication of DNA is not really a good thing. So, how do we get around that? Well, in every single female cell, one of the X chromosomes is transcribing and actively involved in making RNA, which will then go on to be made into proteins. The other X, though, is crumpled into a tight mass that can never be transcribed into RNA. This is an extreme example of DNA packing. And this tight little clump of this chromosome is called a bar body. Barr is the guy who figured this out. This is my favorite visual example of bar body. So this is a female cat. I know it's a female cat because of its coloration pattern. If this was a litter of like 8 kittens with males and females, the males would most likely have been all orange or all black. Okay, so the color of this cat's fur is on the X chromosome. Now, here's the thing with X inactivation is it's random. In some cells, the X chromosome with the orange gene will be relaxed and being able to be transcribed into RNA. This will be the bar body and it cannot be transcribed. In other cells, the X chromosome carrying the gene for the black fur will be relaxed and able to be transcribed, where the orange will be the bar body. Now just look at this cat's coloration. You can see how random this is. Here, the orange gene's being transcribed. Here, the black gene. And that's what gives this cat this tortoise shell appearance. Kind of cool, huh? Even if the DNA is relaxed in chromatin, we still have ways of controlling whether or not that DNA is copied into RNA. We have regulator proteins that have to interact with the DNA, and these are called activators. They are more important than repressors. So what do I mean by that? Transcription is always kind of in the off position. Like if you get in your car is not just going. You have to make it go. Well, same for transcription. So the activators are what will kind of initiate transcription to occur, again, regulating whether or not the gene is going to be transcribed into RNA. Let's take a look at how this works. Okay, enhancers precede a gene. Enhancers are actually DNA. What will happen is these are the activator proteins. So the gene will not be transcribed until these activator proteins bind to the enhancers. Once activator proteins bind, a series of other proteins will bind to those activators, and then eventually RNA polymerase. At this point, RNA polymerase will be bent in such a way that it will directly attach to the promoter and then go ahead and transcribe the gene. Okay. Here's something that's kind of cool. Think about like hemoglobin. That's actually made up of 4 different polypeptide chains. So how does our cell know to make those 4 different polypeptide chains at the exact same time to make hemoglobin? The enhancers have the exact same sequence of DNA. So when these activator proteins show up, they'll bind to all those enhancers and it kind of coordinates that gene expression. Okay, how can we control exactly what proteins are made once we get to the stage of RNA? So the DNA has somehow been relaxed. We have turned the gene on. We've made our RNA. Now what? Well, here's another problem that our cells have. We only have about 20 to 25,000 genes, but we need to make way more proteins than that. One of the things that we can do is called alternate RNA splicing. This controls what type of proteins we'll make, and we can use one gene to make several different kinds of proteins. All right, so what do I mean by that? Here's my pre-mRNA molecule. Remember, I'm going to express my exons and I'm going to put my introns into the garbage. Or I'm going to express my exons and cut out my introns. Right now, I have 4 exons. 1, 2, 3, 4. I will cut out these introns, and I will make a gene with my exons. That's 1, 2, 3, 4. Okay. So what alternate RNA splicing is it's switching out exons for introns and introns for exons sometimes even. So in this example, instead of making a gene that's 1, 2, 3, 4, I'm just going to make 1, 2, and 3, and 4 would then become an intron. Or what if I did this? Sorry, I'm having difficulties erasing. Always fun. Okay, so I make 2 an intron, and 1, 3, and 4 are my exons. You can see I can come up with a bunch of different combinations, and that way I can make different polypeptides from one gene. Once we've taken our DNA molecule and copied it into RNA, specifically mRNA, and sent it out of the nucleus, is there a way to stop it from becoming a polypeptide? There is. MicroRNA and siRNA, or short interfering RNA, both can stop an mRNA molecule from being translated into a protein. MicroRNA will actually bind to complementary sequences on the mRNA, and siRNA can even cleave the mRNA and inhibit translation. So let's talk about RNA interference, or RNAi. These are going to be small RNA molecules, like 20 base pairs long, right here. And what they're going to do is one of two things. They will actually bind to the mRNA molecule and cause it to break apart, or they will bind to the mRNA molecule and act as a physical blocker so that the ribosome cannot translate that messenger RNA into a polypeptide. So in this case, the DNA has been copied into an RNA, but we're going to stop it before it becomes a polypeptide. Again, controlling gene expression. After the mRNA is transcribed and it leaves the nucleus and heads out the cytoplasm, we have even more ways other than the microRNA of controlling whether or not this gene is going to be expressed. One of them is the degradation of mRNA. We talked about this a little bit in the last podcast. Sometimes the mRNA will be broken down right away, especially if it has anything to do with the cell cycle. We'll break it down so that it cannot be translated into a polypeptide. Other times, if it's a gene that we need a lot of, like hemoglobin, we'll actually leave that mRNA to get translated into a polypeptide over and over again. Some research has shown that the hemoglobin mRNA can last up to a month in our cytoplasm, getting translated over and over and over again. And then last but not least, it's how fast we break down a protein. Sometimes proteins like hemoglobin, again, even when it's a full protein, will last for quite a long time, as in months, and some gets broken down right away, controlling how long we express that phenotype. Understanding gene expression has led to a really important laboratory tool called DNA microarrays. This right here is an actual picture of a microarray. So this would be about the size of like a microscope slide. And what you're looking at is whether or not certain genes are being expressed. And we can tell that by kind of the color coding. These are now read by computers, and the computer would tell you, okay, in this cell, these genes are being expressed, these genes are not being expressed. So, like let's say it's a cancer cell. We could see if there's genes on that are causing the rapid cell division, or if there are genes that are off that would stop that rapid cell division. So we can kind of figure out exactly what the problem is. So what you do in a microarray is you actually get the mRNA from a cell. Why do we use the mRNA? Well, because we don't want any introns. We just want exons. And we actually will use like a reverse transcriptase to make what we call cDNA, which actually stands for complementary DNA. A lot of people think that C stands for copied. It does not. All right, and we get that cDNA, and then we can apply that to a cell, and we can figure out I'm sorry, not to a cell, to a slide, and we can figure out exactly what genes are being transcribed, which is an extremely helpful tool if we're looking at a cell that's not behaving as it should. Okay, you guys, that's my presentation on eukaryotic gene expression. I hope you found that useful.